We are delighted to bring you this seminar presented by LabRoots. Welcome, everyone. Thank you for joining us for today's keynote presentation titled Interplay between LRRK2 protein kinase and Rab GTPases in Parkinson's disease. It's my pleasure to introduce you to Dr. Dario Alessi. Dr. Alessi is the professor of signal transduction at the University of Dundee. He obtained his PhD from the University of Birmingham in the United Kingdom and carried out his postdoctoral at the University of Dundee, where he was then appointed the director of the MRC protein phosphorylation unit in 2012. For a complete biography on our speaker, please visit the tab at the top of your screen. If any questions arise during the presentation, we encourage you to submit them in the Q&A box. Our speaker will respond back to you via email. As a reminder, this presentation is educational and offers free continuing education credits. Click on the Continuing Education Credits tab located in the top right corner of the presentation window and follow the process to obtain your credits. Now please join me in welcoming Dr. Alessi. I will now turn the presentation over to him.
Okay, well, thank you very much. I would like to thank LabRoots for the very kind opportunity to give this talk to you. And I'm going to tell you about the work that my laboratory has been doing on this protein kinase termed LRRK2 and its role in Parkinson's disease. So maybe before I get going with my talk, I just wanted to give you a brief background about what I'm interested in. I'm really interested in understanding signaling pathways, how they're organized, how they sense a signal, and how the signal travels, through the signaling pathway to elicit a physiological effect. And, we're especially interested in disruptions in signaling pathways or components of signaling pathways that are linked to human disease. And our hope is that when we understand these pathways in well enough detail, this gives us, clues on how we can play the engineer by working with clinicians and pharmaceutical companies to design potentially new drugs that will restore normality to these signaling pathways that could be, new ways to, better treat or even cure disease, in the future. So the story I'm going to tell you about today, concerns, Parkinson's disease. At the time, I think the story starts in 2004, at the time when I was really working on signaling pathways involved in high regulating human blood pressure and also cancer. And I was very interested in two papers that were published by the groups of Thomas Gasser and Andrew Singleton. They were published back to back at the same time in 2004, where they reported that mutations in an LRRK and a protein kinase termed LRRK2 that I hadn't heard of before, really were causing, a rare form of autosomal dominant Parkinson's disease in the families of patients that, that they had identified. And, this was actually, for me, I remember thinking about the time was really curious and interesting. And I remember thinking, how can a mutation in an unstudied protein kinase really, cause, Parkinson's disease? So, because we were interested in kinases and signaling pathways, we thought we would start a program to understand LRRK2 and how it was regulated and how mutations might, be linked to Parkinson's disease. So LRRK2, it turns out, is one of the largest protein kinases that's encoded in the human genome. It has over 2,500 amino acids. It has several domains. It has an armadillo domain, an ankyrin repeat domain, a leucine-rich repeat, and at the C-terminus has a WD40 domain. But in addition, it also has two catalytic domains. Obviously, it has a kinase domain, but it also has, next to the kinase domain, a GTPase domain that binds GTP and hydrolyzes GTP to GDP. And this GTPase domain belongs to an ancestral family of GTPase domain that's found even in bacteria that's called the ROC core domain. So, in humans, there's sort of 7 really clear-cut pathogenic mutations that cause Parkinson's disease. And interestingly, these are always located in the catalytic domains of LRRK2, either in the kinase domain or the GTPase domain. And the most common mutation in humans is G2019S. So this is why I've highlighted this, in large font. So before I tell you about more about LRRK2, I would like to just give you a very brief background on Parkinson's disease. So Parkinson's disease is the second most common neurodegenerative disorder after Alzheimer's disease. It affects around 10 million people in the world, are affected by Parkinson's disease. And, it's a very debilitating disease that affects, virtually every aspect of people's life who have this, this condition. And unfortunately, all the current treatments that we have for this disease just, alleviate the symptoms, but, don't slow down the progression of the disease. So, and for example, the most widely used drug to treat Parkinson's disease is a drug called levodopa. This leads to, it's converted to dopamine, so mimics the loss of dopamine in the brain. But this is a very old drug. It was discovered over 52 years ago, in 1967. And unfortunately, most of the drugs that have been developed since then are really offer incremental benefits over levodopa. They're just similar to dopamine-producing drugs, but maybe release the dopamine in a different manner. But there's a really very critical need for drugs that slow the progression of the disease down. And our hope is by studying components like LRRK2 that are genetically linked to Parkinson's disease, where we know that the cause of the disease in the patients that have the LRRK2 mutations is an impact on LRRK2 driven biology, then this gives us a unique opportunity to maybe develop the first disease modifying treatments by targeting LRRK2. So I'll tell you a little bit more about LRRK2 and Parkinson's disease. So it turns out that LRRK2 is one of the most commonly mutated genes that causes Parkinson's disease. So if you have Parkinson's disease running in your family, you have around 5 to 10% chance that the gene that will be mutated is LRRK2. So it's actually one of the most common disease causing mutations. And people tell me that in about 1% of sporadic Parkinson's disease patients, there's the LRRK2 mutations are observed. There's 95% or more of patients who have Parkinson's disease have the sporadic form of the condition, in which we don't know what the cause of the disease is, and it doesn't seem to necessarily be inherited from your parents. So the second thing is that the disease progression with LRRK2-mediated Parkinson's disease is very similar to the sporadic form of the disease. So the patients tend to get the disease in their 50s, 60s, 70s, sort of later on in life. This is normally the disease course of the disease. And clinically, there's no way to distinguish the patients with LRRK2-driven Parkinson's disease in terms of the clinical presentations. It's virtually identical to patients with the sporadic form of the disease. And 95% of people with LRRK2 mutations apparently have this G2019S mutation that's located within the kinase domain of LRRK2. So, what's unique about LRRK2 is that all 7 pathogenic mutations induce hyperactivation of the LRRK2 protein kinase, therefore, increases its activity. And this is probably unique in the field of neurodegeneration, that a disease can be caused by, simple activation of an enzyme like a protein kinase. And therefore, this really offers a very unique opportunity to, develop inhibitors that suppress LRRK2 activity to reduce its activity, and as a potential treatment for Parkinson's disease, at least the Parkinson's disease that is driven by, LRRK2 mutations. And many companies in recent years have been working on inhibitors for LRRK2. Potentially the company that's most advanced in this area is a company called Denali Therapeutics, who've recently completed the phase 1A and also a phase 1B clinical trial where volunteers, healthy volunteers, but as well as humans with Parkinson's disease, were given 2 drugs that this company's developed. And the company claims that in these humans they were able to demonstrate that the drugs, the compounds reduced LRRK2 activity by 90%, and most importantly, the compounds were well tolerated when given to the healthy volunteers and also to Parkinson's patients. So, as I said before, LRRK2 might only account for 1% of Parkinson's patients. So an important question is whether or not LRRK2 is involved in patients with sporadic Parkinson's disease. And, we don't really have the answer to this question, but given that LRRK2-driven disease is very similar to the sporadic disease, there's a possibility that at least some patients with a sporadic disease, the disease could be caused by overactivation of the LRRK2 pathway, and therefore this might suggest that LRRK2 inhibitors might benefit patients beyond those just carrying LRRK2 mutations. So this is going to be an important area for future research. So when you work on a protein kinase, the key question is, what is the cellular substrate of this enzyme? And I think LRRK2 is probably one of the hardest problems I've ever worked with. We started working on this in 2004, and it took us till 2016 to identify the key physiological substrates for this enzyme. And I think the reasons for this were because LRRK2 itself has very low catalytic activity, so the substrates, it phosphorylates to a very low level. We also made knockout and knock-in mice that mimic the disease, and we also used chemical inhibitors from pharmaceutical companies. And it's possible that the chemical inhibitors we used from pharmaceutical companies had a lot of off-target effects. So when we did the mass spectrometry, we saw lots of hits beyond the true substrates. Also, when you generate an LRRK2 knockout mouse or an LRRK2 knock-in mouse mimicking the disease-causing mutations, you probably disrupt many pathways, in a cell and tissues. And over time, you get rewiring of signaling pathways to compensate for the loss of LRRK2 or the disruptions in the LRRK2 pathway. And therefore, you see many indirect effects that also mask that needle in the haystack from when you do the genetic experiments. So to overcome this problem, what we decided to do was to generate an inhibitor-resistant mutation, that made LRRK2, maintained its normal catalytic activity, but made it resistant to the chemical inhibitor that we're using. There was a compound termed MLI2 that was provided by the Merck pharmaceutical company. So we identified a mutation in LRRK2 that made LRRK2 resistant to the Merck inhibitor, and then we made a knock-in mouse that mimicked this mutation, and then we made cells from this knock-in mice that we could study. And here, the knock-in cells will be identical to the wild-type cells because the mutation we chose doesn't change the LRRK2 activity. And the only difference that occurs is when you add the inhibitor to both the wild-type and the knock-in cells, then you're looking for things that are inhibited in the wild-type cells, but not the inhibitor-resistant knock-in cells. And by doing these very laborious experiments, and then the mass spectrometry for these experiments was undertaken by Martin Stiger, and who was based in Matthias Mann's laboratory. The key set of substrates that were identified were a small group of Rab GTPases, where we were able to demonstrate that LRRK2 phosphorylates these enzymes right in the middle of the switch 2 binding motif. This is the area here that's shown in blue. This is the key business end of the Rab GTPase that binds to physiological effectors. And and so Rab proteins are really very fundamental proteins that regulate, all aspects of, cell biology, all vesicular trafficking, membrane integrity. Is controlled by a group of about 70 Rab GTPases, so I just wanted to show you, and they all work by binding to, GTP and they're hydrolyzing GDP to GDP, and they're in their active form when they're bound to GTP, and they can interact with, effector proteins by using this switch to, docking interaction sites. So I just want to show you some data to really demonstrate that, that Rab GTPases like Rab10 are indeed physiological substrates for LRRK2. So here we use a gel that separates out, it's called a phosphate gel, that separates out the phosphorylated Rab from the dephosphorylated Rab. And so in the upper band, you can see a few percent, maybe 2% of the Rab protein is phosphorylated. And when you add the MLI2 LRRK2 inhibitor, when you get to a dose of 3 nanomolar inhibitor, you can see that the Rab protein is dephosphorylated. And then we did the same experiment with the, these A216T knock-in cells. These are the cells that are now resistant to the MLI2 inhibitor, because this mutation, reduces the ability of LRRK2 to be inhibited by MLI2. And you can see here now you have to go to 100 nanomolar inhibitor to induce the dephosphorylation of the Rab protein. So, this really persuaded us that the Rabs were the key substrates for the LRRK2 pathway. And then the next question was whether the disease-causing pathogenic mutations would enhance the Rab protein phosphorylation, because you would think of the Rabs were the relevant substrates for LRRK2, that the knock-in mutations that cause Parkinson's disease would enhance the phosphorylation. And then this is what we found. For example, this is the R1441G mutation that's present in the GTPase domain. When we get cells with this knock-in mutation, you can see you get a 3 to 4-fold increased levels of Rab10 phosphorylation. And when you add the LRRK2 inhibitor, in this case, we're using a GSK compound, you induce complete dephosphorylation of the Rab protein. So these experiments suggested that, Rabs like Rab10 were the physiological substrates, and the disease-causing mutations all led to an enhancement of the Rab activity, of the Rab phosphorylation. And so it turns out that there's this phosphorylation site we identified in the Rab GTPases. It's actually found in about 50 of the 70 Rab proteins. So next question was, how common is, which subset of the Rab proteins are phosphorylated by LRRK2? So we therefore had to test very carefully all 50 Rab proteins that had a phosphorylation site in this particular sequence in its switch 2 motif. And then for those that we first tested this in an overexpression experiment in 293 cells, and for those that we found were phosphorylated, we then did secondary experiments to look at the endogenous proteins. And thus far, we think that there's about 13 Rab proteins that are probably genuine physiological substrates for LRRK2. So this is Rab1s, the Rab3s, the Rab8s, the 10, 12, 29, 35, and 43. And for these Rabs, we have very good evidence that the endogenous protein is regulated by LRRK2. So when you knock it out, you lose the phosphorylation, or when you inhibit it, you lose the phosphorylation. But when you have a knock-in mutation that mimics the Parkinson's disease-causing mutation, you enhance the phosphorylation of these Rab proteins. So the next stage of the work was to use to generate monoclonal antibodies that would enable us to better recognize and detect LRRK2 phosphorylated Rab proteins. These are really the beacons of LRRK2's pathway activity, in tissues and in cells. So it's very critical that we can identify these sites and measure them accurately. So we spent a lot of time working with the Michael J. Fox Foundation and ABCAM, and we've generated a long list of antibodies. I think we're approaching over 30 antibodies now that we've identified that can detect and immunoblot, immunoprecipitate, and can be used for immunofluorescence, as well as also FACS analysis to study the LRRK2 phosphorylated forms of the Rab proteins. And these are wonderful reagents. For example, the antibody that picks up phospho-Rab10, if you use a cell like a fibroblast cell, you can see you only have one major protein that's detected in the whole extract that goes down when you add an LRRK2 inhibitor. And we can use these reagents to study more globally how pathogenic mutations affect Rab protein phosphorylation. For example, we found that the G2019S mutation in knock-in mice gives around a 1.8-fold increase in the Rab protein phosphorylation, whereas mutations in the R144.1 residues give around 3.5 to 4-fold increase in Rab protein phosphorylation. And when you use LRRK2 knockout cells, you see no Rab protein phosphorylation. And when you use kinase inactive knock-in meths that were provided to us by GSK, you also see no phosphorylation of the of the Rab protein. So as I said before, we've made a lot of these different antibodies, and I'm not going to go through all of them, but a lot of them that we're working on have been made, are listed here. And we think that these will be very important reagents for the field to study LRRK2 pathway biology. And we've been very careful to make sure that the antibodies are immediately released through these commercial companies as soon as they're characterized, so that the whole field, including pharmaceutical companies, can benefit from from this work. So another next question for us is, okay, we know that LRRK2 phosphorylates these Rab proteins in the middle of this effector binding motif, but how is this going to affect the next stage of the pathway, downstream of the pathway? And and it seemed to me that since only a few percent of the Rab protein was phosphorylated, the most likely way that this would work would be that the phosphorylated Rab would act as a receptor for a new set of proteins that can only interact with the LRRK2 phosphorylated form of the Rab protein. So we conducted a lot of work to identify proteins that might only bind to the LRRK2 phosphorylated form of the Rab proteins. At my lab, and also the lab of Matthias Mann, also run by Martin Stiger, did a lot of mass spectrometry to look for such receptors for phosphorylated Rab proteins. And thus far, we've really focused on Rab8 and Rab10, and we've identified 4 proteins called RILPEL1, RILPEL2, GIP3, and GIP4 that have a RH2 domain in the sequence that we've been able to show binds very selectively to the LRRK2 phosphorylated form of the Rab proteins. And this and other work we've done suggests the following model of how this pathway might work. So when the Rabs are made, they obviously they live on membranes, but originally they can move between membranes by forming a complex with this GDI protein. It's a chaperone protein that binds to most Rab proteins and sort of solubilizes these Rab proteins so they can be moved between membrane structures in the cell. And then they'll be taken to a membrane where they'll sit on the membrane, and the prenulated C-terminus that's shown with this squiggle is critical for maintaining the Rabs on the membranes. And the Rabs will be inactive because they'll be bound to their GDP-bound form. But in response to a signal or an agonist, this GEF protein here will come along and switch on the Rabs by converting it into the GTP-bound form. This GTP-bound form then will bind to many effectors to regulate normal membrane or vesicle biology. But what happens when LRRK2 comes in, LRRK2 will phosphorylate the Rab at this switch 2 motif. and this will block its ability to bind an effector protein, because most of the effectors we've tested can no longer bind to the Rabs once they're phosphorylated. And therefore, these effectors will dissociate from the Rab proteins, and therefore the phosphorylated Rab protein at first will be completely naked and trapped at the membrane that is phosphorylated, because it can't bind to GDI, so it can't go to another membrane in the cell. But then what happens is that these other effector proteins that can only bind to the phosphorylated effectors like RILPEL1, RILPEL2, GIP3, and GIP4, come in. They can bind to the phosphorylated Rabs and trigger distinct downstream biology. So obviously a key part of our projects now is to really understand how these effector proteins regulate Rab biology. And we think we've only identified 4 of them, but we think there'll be many more, and we're setting up screens to identify novel effectors that will bind to the LRRK2 phosphorylated Rab proteins. And in collaboration with the laboratory of Amir Khan and a postdoc in his lab, Didier Woesboots, we've been able to crystallize phosphorylated Rab8 bound to the RH2 domain of RILPEL2, and the structure is shown here. And what's quite nice is you can see specific residues that are involved, such as the arginine 132 residue in RILPEL2, that's really critical for coordinating interaction with the LRRK2 phosphorylated Rab8. And when you mutate these residues, you lose the ability of RILPEL2 to bind to phosphorylated Rab8 protein. And my great collaborator Susan Pfeffer, who works at the University of Stanford, has done some beautiful work to study the role that this interaction between RILPEL1 and Rab10 plays in cells. And her laboratory have shown that this really sort of plays an important part, plays an important role in the pathway that regulates ciliogenesis. And indeed, when you have LRRK2 pathogenic mutations, she was able to demonstrate that this leads to an inhibition of ciliogenesis, and this can be restored by inhibiting LRRK2 or the ciliogenesis come back. And this pathway really does require the interaction of RILPEL2 binding to phosphorylated Rab10 to get this inhibition effect on ciliogenesis triggered by LRRK2. And the Pfeffer lab was able to show that in especially in the striatum and the cholinergic neurons in the brain, that this inhibition of the ciliogenesis pathway, it leads to a reduced response to sonic hedgehog signals, and this in turn reduces the neuroprotective GDNF signaling that comes back to the dopaminergic neurons, and this could really underline potentially the increased death or loss of or degeneration of dopaminergic neurons, and potentially links LRRK2 to Parkinson's disease. So there's more work to be done here, but this is a sort of one model that explains how LRRK2 might be linked to Parkinson's disease. Then our next question is, what is upstream of LRRK2, and how is it regulated? And to do this, we've really thought that maybe one way to start was maybe mutations in one of the other 20 or so genes that cause Parkinson's disease. Maybe mutations in these genes could also switch on LRRK2 and increase Rab10 phosphorylation, and this could be the mechanism by which these mutations are linked to Parkinson's disease. So what we're doing now is we're testing many of these mutations to see whether they influence Rab10 phosphorylation. A few years ago we first studied Rab29, and this work's all published. So and this is also a collaboration with Susan Pfeffer's lab, and I should say that the laboratory of Andrew West and Mark Cookson and the laboratories in Japan got very similar results, where we all showed that Rab29 is located at the Golgi network, and it somehow interacts with the N-terminal domain of LRRK2. So especially when LRRK2 has the pathogenic mutations in the GTPase domain, that actually increase GTP binding. The LRRK2 is recruited to the Golgi where it becomes activated. And you can see the activation by looking at the enhanced phosphorylation of Rab10 or the phosphorylation of itself at serine 1292 or Rab29, that's phosphorylated at threonine 71. And, this is a very robust result that you can see very clearly with overexpression studies at least, but more work is still really needed to understand how Rab29 interacts with LRRK2 and especially how this activation occurs. There may be other components that might be needed at the membrane to achieve this, because so far it has not been reproduced in an in vitro experiment in the test tube. The second component we studied is this VPS35 component that VPS35 it's a very critical core component of a complex called the retromer complex that's made of 3 subunits. It's got VPS35 here shown in red. It's got a second component called VPS29 and the third component called VPS26. And this acts as a critical cargo binding and shuttling component that takes cargo from the endosomes and small vesicles to the Golgi, and also it takes things from the endosomes to the plasma membrane. And so it plays a very critical role in in cargo shuttling in this cell. And what's interesting actually is that the cargo, I've marked in the diagram where the cargo bind typically to the retromer complex, but you'll see on my slide there's an aspartic acid 620 residue that's highlighted, and this residue is in a region of the retromer complexes whose function isn't known. But when this residue is mutated to an asparagine residue, this causes very severe autosomal dominantly inherited Parkinson's disease. And patients with this mutation tend to get the disease at least 10 years before patients with LRRK2 mutations. And it also seems to be more penetrant. It seems that virtually every patient with an AS620N mutation in VPS35 develops Parkinson's disease, whereas this isn't necessarily the case for LRRK2. So we were interested to see whether or not this VPS35 620N mutation, could affect LRRK2 activity or Rab10 phosphorylation. So we got hold of a knock-in mouse that had been made with this mutation. I think it was originally made by the Michael J. Fox Foundation and Matt Farah's laboratory, and we made cells from these mice. And what we found was that we got the same results in the cells and also tissues of the mouse that we looked at. We saw a very dramatic enhancement in Rab8, Rab10, Rab12 phosphorylation in the cells that had this VPS35 620N mutation. And when you add the LRRK2 inhibitor, this phosphorylation goes away, telling us that the mutation is leading to the enhanced phosphorylation of these Rab proteins. We don't know the mechanism here. One mechanism is it could be that this mutation is activating LRRK2, so LRRK2 is more active, and therefore phosphorylates its Rab substrates more effectively. Or it could be somehow bringing the Rabs and the LRRK2 together, or putting them together in the same vesicles more efficiently than the non-mutated form of VPS35. And if you put LRRK2 and the Rabs together in the same vesicle, maybe you'll get enhanced phosphorylation. But I should also say that the increase in Rab phosphorylation we see in these cells and also tissues from the mice is much more than we see with LRRK2 pathogenic mutations, which give the most 3 to 4-fold increase in Rab phosphorylation, whereas the VPS35 is giving us 5 to 8-fold increased phosphorylation. So it's possible that this explains why if VPS35 patients with these mutations maybe develop Parkinson's disease earlier, and it's more penetrant if the LRRK2 activity is more elevated. in these patients. So, then we were also interested to see whether we could interrogate LRRK2 activity in patients with VPS35 mutations. So this project was largely driven by my clinical collaborator, Esther Samler, and Francesca in my laboratory, and we also collaborated extensively with Alexander Zimpritz at the University of Vienna and his colleagues. And Alexander has a group of around 20 patients who have these VPS35 mutations. And we just really wanted to ask the question is, could we demonstrate in these people that they had enhanced Rab protein phosphorylation? So to do this, we developed an assay using monocytes or also neutrophils that we isolate from the blood of these patients. Obviously, the patients with the mutations have the mutations in every cells, but and then we just wanted to test whether this mutation in the monocyte in the LRRK2 would lead to an enhanced Rab protein phosphorylation. So these are the experiments that we did in monocytes. We had as a control a number of idiopathic Parkinson's patients, and we liked those as the control because we knew these patients didn't have mutations in VPS35 or LRRK2. They're the same age as the patients with the VPS35 mutations, and they're on the same medication. And then when we purify the monocytes, we treat them plus and minus an LRRK2 inhibitor, and then we lyse the cells, and we blot for phospho-Rab10, and we also blot for total levels of Rab10. So if you look at the total levels of Rab10, it's quite remarkable. In each of these donors or individuals, you can see the levels are very, very constant. If you look at levels of LRRK2, they're very constant, and the levels of VPS35, 26 are also very constant between the different individuals. And in the idiopathic patients, you can see significant Rab10 phosphorylation, and when you add, as you expect, the LRRK2 inhibitor, the phosphorylation goes down to background levels. So this proves to us that we are measuring LRRK2-mediated Rab10 phosphorylation. But what's quite striking, you can see here the three idiopathic patients, the three patients with the VPS35 mutations, you can see a 3 to 4-fold enhancement in Rab protein phosphorylation that goes down with the LRRK2 inhibitor, suggesting that we see the same results in patients with VPS35 mutations as we do in a mouse model. And I think Esther and Alexander have now analyzed, I think, nearly 20 patients with VPS35 mutations, and we see in all these patients this very robust elevated Rab10 phosphorylation. And then this assay is now being widely used to interrogate LRRK2 activity in humans, and it's a very simple way to assess whether a mutation or another disruption in biology is increasing Rab10 phosphorylation in a human patient. So I'll now go on to before I finish with VPS35, I just wanted to highlight that we've been working with Stressmark Biosciences, and because some of the VPS35 antibodies we were using weren't very high quality, so we've developed two fabulous antibodies that pick up VPS35 that are listed here, and they're validated because when you knock out the VPS35 using CRISPR methodology, you lose recognition of the VPS35 protein. And we've also validated these antibodies work immunofluorescence. So, in the wild type cells, you can see these very red puncti located around the cells. These are the small vesicles that the VPS35 is located in. But when you knock out the VPS35, you lose these puncti, these vesicular structures. So we've tested every single available VPS35 antibody, and then these two antibodies are vastly superior in our opinion than any other antibody that's been developed. So I would recommend if you work in VPS35 that you test these antibodies. So as I said before, we don't know the mechanism by which this mutation in VPS35 stimulates Rab10 phosphorylation or Rab protein phosphorylation. This is really an important area of research in our lab, is to try to, work this conundrum out. So the next part I'm going to tell you about is, the role of LRRK2 plays in the immune system. So it turns out that LRRK2 is most highly expressed in humans in immune cells. It's much higher in monocytes, neutrophils, macrophages than, for example, than it is in neurons. In the brain, it's also quite highly expressed in astrocytes, microglia, and oligodendrocytes, but it's normally quite low in neurons. And so therefore, because of this expression pattern, people wondered whether it had a role to play in protecting, the people from opportunistic infection by working in the immune system. And genetic analysis has shown that GWAS analysis has linked mutations in LRRK2 to inflammatory conditions, including ulcerative colitis, Crohn's disease, and also susceptibility to leprosy. And other studies in mice have shown that mutations that stimulate LRRK2 activity, protect against listeria and salmonella infection, and vice versa. When you knock out LRRK2, the mice are more prone to these infections. So question is whether when you infect an immune cell, or immune cell recognizes a bacteria or a pathogenic fungi, does this impact the LRRK2 signaling pathway? So we've done some work with the pathogenic bacteria, but the results that I'm going to tell you about today are the work we've been doing with fungal pathogens, and especially the Candida and Aspergillus. That's a very common, fungal pathogens that affect people, especially people who have, problems with their immune system that are compromised. And this work's been a work with my collaborator Simon Arthur's lab and Stephanie Laber and Ivona Fan in my lab, who have been working on this project. And what we found is when we take these are mouse bone derived macrophages, and we infect them with Candida albicans, for example, and this can be done even at an MOI of 1. You see that within 30 minutes, you see a very robust increase in Rab10 phosphorylation that stays high for up to 4 hours. The cells start dying around 4 hours, but it stays high up to that point. And then we know that this is mediated by LRRK2, because when we add the LRRK2 inhibitor, the phosphorylation of Rab10 is maintained low. So this suggests that, like the VPS35 mutation, Candida infection is somehow bringing LRRK2 and Rab proteins together, potentially on the same vesicle, and promoting Rab10 phosphorylation. And it seems that for this to happen, the Candida has to be alive. So if you heat kill them, or you in other experiments you've used the cell wall, or we don't see this stimulation of Rab10 phosphorylation following infection with the heat-killed Candida albicans. And we've seen the similar results with Aspergillus, that infections also stimulate Rab10 phosphorylation by LRRK2, but if you use yeast cerevisiae, you don't achieve this. So work is ongoing in my lab to really work out, what are the key mechanisms that bring LRRK2 and Rab proteins together following infections. And then obviously the most important question is, how important is this pathway in protecting patients from, infection? So, it's quite interesting. The model is that if you have an LRRK2 pathogenic mutation, you might be more protected from against opportunistic infections. And then this would have been very important hundreds or even thousands of years ago, when people obviously didn't live much past their 20s. That if you had a mutation that protected you from developing infection, that sort of primed your immune system, this would actually be beneficial. And but the problem with this is when you have a primed immune system that might be good for you when you're young, as you get older, this might increase the chances of maybe leading to neuroinflammation, or and that might be promoting Parkinson's disease. So this is a concept is called antagonistic pleiotropy, something that might be good for you when you're young, but then becomes detrimental for you as you get old. And many sort of signaling pathways that cause disease might be doing this, sort of protecting people when they're young, when they're young, but therefore, the activation of the pathway throughout life becomes detrimental. So the next thing I'm going to talk to you about is the protein phosphatase that acts on the Rab protein to dephosphorylate the Rab protein, after LRRK2 has put the phosphate on. All protein phosphorylation in cells is normally phosphorylate, is reversible, and the phosphatases work to dephosphorylate the protein. So and we think that the Rab phosphatase is very active in cells because when you treat cells with an LRRK2 inhibitor, you can see within 1 minute the Rab protein is becoming substantially dephosphorylated and is sort of completely dephosphorylated within about 5 minutes. Whereas if you look at another phosphorylation site in LRRK2, a site called 935, here it takes about 80 minutes for it to become dephosphorylated. So, we're very curious to understand what is this very high level of phosphatase activity that's working on the Rab proteins. And obviously, this enzyme is potentially very important because this will keep Rab protein phosphorylation low in cells, and if you inhibited it, the levels of Rab phosphorylation could potentially increase to toxic levels. So it turns out that there's little work going on in the world now on protein phosphatases. Most of the work on protein phosphorylation focuses on the kinases, but we now know that there's 189 catalytic subunits of phosphatases. They lie within 10 different phosphatase families. Many of these catalytic subunits interact with regulatory subunits. So there's literally probably hundreds, if not thousands, of components in the cell that regulate protein dephosphorylation. And this is a vastly understudied field of research. So we undertook an I won't go over the details, we undertook an siRNA screen to knock down all of these phosphatases individually and also some known regulatory subunits. And for each of the knockdowns, we tested the impact that this had on Rab protein phosphorylation in the cell. And when we did the screen in two different ways, one where we just looked at the dephosphorylation of the phosphorylation status of the Rab protein after the siRNA screen. In the second screen, we did the siRNA screen for 72 hours, and then we incubated cells for 5 minutes with an LRRK2 inhibitor, and we looked for phosphatases when we knocked down maintained high levels of Rab10 phosphorylation. And then the top hit in this screen was an enzyme that is called ppm1H. It's a very poorly studied phosphatase. It belongs to this family of phosphatase that's known as the ppm of magnesium-dependent family of phosphatases. In humans, we have 20 members of this family, and ppm1H belongs to one of them that is shown here. It has two relatives, J and M. And it's a very simple phosphatase. It has a phosphatase catalytic domain, and it's only got 514 residues. It doesn't seem to have any other, obvious domains in its structure. And, to validate the siRNA screen, we made CRISPR knockouts of ppm1H. We actually made 10 different CRISPR cell lines, some that remove all the protein, some that remove the upper splice, the upper form of ppm1H. But what we found is that in all 10 ppm1H knockout clones, we resulted in an enhancement of Rab10 phosphorylation that was seen in these cells. We also found that when we overexpress ppm1H in cells, we dramatically inhibited Rab10 phosphorylation. So in these cells, when you overexpress catalytically inactive phosphatase known as H153D, you maintain high levels of Rab10 phosphorylase, but when you overexpress the wild type inactive phosphatase, you dramatically reduce the degree of Rab10 phosphorylation. And we see the same results with a lot of other Rab proteins, not just Rab10. So we think that PPM1H is quite non-selective. It seems to dephosphorylate, a large group of Rab proteins that are phosphorylated by LRRK2. And we think this is a direct effect, because when we take phosphorylated Rab10 protein and we incubate it with a wild type PPM1H, we can see a time-dependent dephosphorylation of Rab8, where this is an in vitro experiment suggesting that the PPM1H works directly on the phosphorylated Rab protein. But when you do the same experiment with catalytically inactive Rab proteins, you don't see dephosphorylation. And one advantage of phosphatases is that working with phosphatases, you can actually generate what's known as a substrate trapping mutant, and you can design the mutant that actually binds to your phosphorylated substrate, but can't really dephosphorylate it and becomes trapped, onto the phosphorylated substrate. And this is a very good method for validating physiological substrates for your phosphatase, because if you can make a substrate trapping mutant, you can then isolate the substrate trapping mutant from cells, and you can see what else is bound to it. And these are normally sort of the physiological substrates of the phosphatase. So we identified the substrate trapping mutant of PPM1H, and this is a mutation called D288 alanine. And you can see when we express this substrate trapping mutant of PPM1H in cells, this is the D288A mutant, and we immunoprecipitate this from cells, you can see that it binds to very high affinity to endogenous phosphorylated Rab8 and Rab10. Whereas if you overexpress the wild type or another catalytically inactive mutant of PPM1H, it's not a substrate trapping mutant, it doesn't interact with it. So we think this provides the strongest evidence that the Rab proteins are genuinely physiological substrates of the PPM1H. So much work is still going on. We really want to understand more about how PPM1H activity is regulated and controlled. We're interested to know whether the mutations in PPM1H that might increase its expression, maybe protect people from Parkinson's disease or vice versa. If you've got an inhibition of PPM1H, would this lead to an increased chance of getting Parkinson's disease? And, is it possible that, to develop compounds that enhance PPM1H activity, could these potentially be, new agents to better treat, Parkinson's disease? So in the last few slides, I'm going to tell you about, some work that we've been doing to better interrogate LRRK2 in humans, and some mass spectrometry analysis that we've been doing to try and develop more sensitive and quantitative assays than the immunoblotting systems that I've been discussing. And then to do this is again led by Esther Samler, together with the laboratories of our collaborators. In this case, is from Barcelona Hospital, from the lab of Eduardo Toloso and his colleagues. And we've been able to get hold of numerous patients with G2019S mutations, and also 5 patients with the R1441G mutation. And by taking neutrophils from these individuals, as I mentioned before, we were able to assess Rab10 phosphorylation in the neutrophils derived from these people. And when we do the experiments with the 5 people with the R1441G mutation, and this includes 3 carriers and 2 patients with Parkinson's disease, you can see in all cases we can see a robust enhancement of the Rab10 phosphorylation compared to the control donors that we've used for these patients. And then we've done the same experiments for patients with the G2019S mutation. But in this case, this mutation doesn't seem to increase Rab10 phosphorylation to a big enough extent that we can visualize this on an immunoblot analysis. And this data is shown in this slide here, where we can find that, with the R1441G mutations, it's easily seen that there's enhancement in Rab10 phosphorylation, but with the G2019S patients, there's really no significant increase in Rab10 phosphorylation over the controls, unfortunately. So because of this, we decided to develop more robust mass spectrometry technologies to look at this. So here, this is a paper which is done by our collaborators Matthias Mann and Ozge Karayel and their colleagues at our laboratory helped to a small extent, and you can read the methodology in this paper. But it's a very robust method to study the stoichiometry of Rab protein phosphorylation. So and it's much more sensitive and quantitative than the immunoblot analysis. So we've reanalyzed all these samples with this more quantitative mass spectrometry assay. But here again, we see the same results as we saw with the immunoblot analysis, that it's not easy to see an increase in Rab10 phosphorylation from the samples derived from the G2019S carriers or patients, but we can still easily see this increase in the R1441G derived patients. So I think that's shows that the assay works, and the immunoblotting assay is not too bad either. And then in the last 2 or 3 slides, I just want to tell you about a new assay that my laboratory has been setting up. This is a multiplex assay that enables us to study the phosphorylation of Rab 1, 3, 8, 10, 35, 43, LRRK2 total levels, as well as the phosphorylation sites of 9, 10, and 935 on these sites. And to do this, we immunoprecipitate. We take cells or tissue extracts. We digest the whole protein in these extracts to tryptic peptides, and then we immunoprecipitate these tryptic peptides with a group of antibodies that IPs, the key peptide antigens that are required to assess the levels of these proteins. We also spike the extract with aqua heavy label peptides for each of these reporters, and then we are able to do targeted, high resolution mass spectrometry to assess the levels of these peptides in the samples. And by doing this, we can now very easily assess phospho-Rab levels and LRRK2 pathway indicators in cell extracts in a much more rapid way and quantitative manner than can be achieved by doing immunoblot analysis. And this is just one example of the mass spec assay shown at the bottom versus the immunoblot analysis. And then you can, with the advantages of the mass spectrometry assays, you don't just measure Rab10, you measure these 14 other readouts that I discussed, and you can see very clearly that other Rab proteins are affected by LRRK2 mutations, and we think this assay is going to be heavily used in the future to assess the LRRK2 signaling pathways. So I think I'm nearly run out of time now, so I think I won't go through the rest of my data. I just want to conclude by telling you that the pathway we've been working on is, I think we started to work out the nuts and bolts of the LRRK2 pathway. It regulates Rab protein phosphorylation mutations in Rab29 and VPS35 feed into this pathway, also with infection and maybe other environmental pathways. And then the phosphorylated Rab proteins, these seem to be the element that's probably linked to the Parkinson's disease. And then I've also told you about the work of Susan Pfeffer's laboratory in which ciliogenesis is linked to Parkinson's disease through this RILPEL1 signaling pathway There's much other work in the field that's been done suggests LRRK2 regulates lysosomal stress and synaptic vesicle trafficking, and this presents a mechanism by which LRRK2 can be linked to Parkinson's disease. And there's also this third link with the immune system by controlling neuroinflammation could be the third way by which LRRK2 is linked to disease. And then other work in the field has demonstrated that Rab biology is likely to lie at the heart of understanding Parkinson's disease. There's work shows that the PINK1 protein kinase that's also implicated in Parkinson's disease regulates Rab phosphorylation at a distinct site to LRRK2. The work of the late Susan Lindquist has shown that alpha-synuclein's ability to be toxic in cells, and cause, cell death is linked to its ability to interact with Rab proteins, mutations in 39B, Rab39B, and other proteins like VPS35, VPS13, and DNA JC6 that are also linked to Rab proteins. So, so there's much more work to be done to understand the biology of Rabs. This is linked to Parkinson's disease. And I just want to end by acknowledging the large group of people who've been involved in this work. In my lab, it's been the work of Francesca, Esther, Elena, Raja, Rafik, Assad, Pavel, Ying, Kieran, and many others over the years that have been involved in this work. And we've had always, for the last 5 or 6 years, very strong and beautiful collaborations with the laboratory of Matthias Mann and increasingly Susan Pfeffer at the University of Stanford. Our work's been supported tremendously by the Michael J. Fox Foundation right from the beginning when we started working on LRRK2. And, this collaboration was critical for success. And we've also received funding from the Medical Research Council of the UK and Parkinson's UK. And I'd like to thank our other clinical collaborators, including Alexander Zimpritz, for providing reagents. And thank you for your attention.
Thank you, Dr. Alessi, for that outstanding presentation. For additional informative presentations, view our agenda on our events page. As a final reminder, our speaker will follow up with any questions you've submitted via email. Thank you again for your participation.